So today I'm going to talk about B cell development. The set of slides that I posted are for both today and for Wednesday on B cell development and selection because really it's one long continuous process. So that's what we are going to talk about. So recall that we've been thinking about VDJ sort of at this level of thinking about what's going on with the DNA. And so we've seen kind of what's happening with the gene segments in the light chain and the heavy chain, as well as sort of specifically what's happening at different pieces of the genome. You can also see here, this figure sort of compares the germline DNA and then the rearranged DNA, where we've gotten rid of some of the intervening sequences. You can see we also have listed the primary mRNA transcript and then the actual mRNA, where we will have, by the time we get to the final mRNA, we've basically removed all of the parts that are not coding for our antibody. And these are changes that are happening at the RNA level by splicing. So you can see, like, there's all of this DNA that doesn't code for our antibody right here. It's in the first transcript, but in the final transcript, it's not there anymore because there's been some splicing. And then that spliced mRNA will get translated into our antibody. So we left off last time talking a little bit about the junctional diversity processes and P and N nucleotides. And I just wanted to give you a couple of other examples to help you understand these processes. So here, what you can see is I have a V segment and a J segment. It's a light chain, A, because light chains go with B and J with no D, but also because there's an L. And so the V segment and J segment are in black. And then the RSS is in red. You can see I've got an RSS with a 23 base pair spacer, an RSS with a 12 base pair spacer, and I've got my heptamer spacer and nonomer. So, and where we are right now is that RAG has basically grabbed onto my two RSSs and sort of helpfully put them like this. And so the first thing that's going to happen is RAG is going to form a nick. And so it's going to cleave one strand of DNA, say like right there, or right here, while leaving the other strand intact. And so what will eventually happen is that you will get something that looks like that. Note that I'm only drawing one of them here just to make our lives a little bit easier and to make the slide not quite so busy. So I'm only drawing the V. The events I'm showing you here would happen at the J as well. It's just that's a law of drawing. So here you can see now we have the bottom strand has stayed the way that it was before. The top strand is broken because of my DNA nick, and it has a free 3' hydroxyl. Now that free 3' hydroxyl is going to perform a nucleophilic attack. So it's going to attack this A. And when it attacks that A, we're going to break the rest of this RSS off. So it's going to be missing. See, now it's missing. And the T is bound to the A. So we have this new hairpin that's bound. The other piece of DNA is off to the side somewhere else, being a signal joint that we don't need right now. And so hairpins are weird. We have to do something about them before we can continue with the DNA. And the thing that happens is we have Artemis, that enzyme that's going to open the hairpin. Artemis is going to act in this area, but Artemis is going to be imperfect and could act on the DNA in a few different locations. So Artemis could cut here, but it also could cut here, 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 it basically can go up to 5 base pairs in either direction. So it's got a bunch of choices. So I'm going to show you the example of what happens if Artemis cuts right here. If Artemis cuts right there, that's what the top strand is going to look like, because these base pairs down here are going to kind of get swung up to the top. And be part of this strand now. While we'll leave these remaining. And so now we have this weird little overhang. And polymerases that are part of the DNA repair enzymes are going to fill this in. And so we'll fill this in. Like that. If you look at this, you notice that some of this is what we started with. And so the part that I put in the box is the same as what we started with. So I might call this the germline BL sequence. And then these other base pairs are newly added. They were not in the original DNA. And they are all P nucleotides. And the way I can tell that they're P nucleotides is I can look at this A and be like, oh my gosh, this A could have been bound to this T. They match. So that could be a P nucleotide. And this T, oh my gosh, it could have been bound to this A. And I can go through and look and see, is it a P nucleotide? If TDT then came in and started adding N nucleotides or random nucleotides, it could just add whatever it felt like. So there's 4 A's. No reason, just because we picked A, and that would also get filled in. And so those would be N nucleotides. The other ones would be P nucleotides. And so this is a past question I have asked. I put 2 of these in the slides. I couldn't decide if I wanted to go through 2 of them or just 1 of them. I figured if I'd had the slide, I could always just skip it. And so you have a question like this on your problem set, and so I want to talk through how in the heck we would figure this out. What this question really, the things you really need to know to answer this question are, what are P nucleotides and what are N nucleotides? So you need to know kind of like where they come from. And you also need to know that the steps of the process are P nucleotides happen first, and then N nucleotides happen after. And so knowing those 2 things will help you address this question. And so I'm telling you earlier in the question about a B cell, and I show you the germline sequences of the J and the D, kind of what we started with before. And then I actually look at a B cell that's done rearrangement. So those are the befores at the top. This is the after. And I ask you to label the origins of these nucleotides. So we've got a whole bunch of base pairs, and I have to tell you where those base pairs came from. And there are a couple things that we should do. So the first thing I can do is I can look at the befores. And I can notice that this part of the before matches the stuff above. I was lazy, and I liked my cat that day. And so the origin of this is that it was IgDH2. And so now all those base pairs, I'm done. I can also go over to this other side and be like, yeah, this was JH3. So now I got 4 left. So originally I had this many base pairs I had to figure stuff out about. I only got 4, much easier. And I'm going to deal with those 4 individually. So I'm going to sort of make a decision about each one. And so we have to remember that the steps go, we add P nucleotides before we add N nucleotides. So if there are P nucleotides, they're sort of closer to the gene segment than the N nucleotides would be. And basically what we're going to do is we're going to look on each side, either next to the D or next to the J, and say, are these base pairs that are next to you P nucleotides? If we find some P nucleotides, we're going to say, yes, it's a P nucleotide. If they're not P nucleotides, then what are they? They must have been N. So we're basically just going through and saying, are you a P nucleotide? After we figure out which ones were P nucleotides, we just know all the rest of them are Ns. And so the first thing I'm going to look at is this A. If this A had been a P nucleotide matched to the D segment, could this be a P nucleotide that came from this side? So I'm hearing some yeses. How do you know it's a yes? Yeah. So A can go with T. So yeah, that could be a P nucleotide, right? All right, P nucleotide. Done. Now we got 3 left. All right, this A. Could this A be a P nucleotide over here? No, because if it had, it would be A matching with A. A doesn't match with A. So not a P nucleotide on that side. Before I call anything an N, I'm going to try the other side. So let's look at this one. This C. Could this C be a P nucleotide on this side? Yeah, it could, because C goes with G. Okay? So I know that one's a P nucleotide. All right. How about this G? Could this G be a P nucleotide on this side? No, because that would have been G with G, and that's not right. So clearly I'm done with the P nucleotides. Because again, we remember the steps are P nucleotide then N. So if that one's not a P nucleotide on this side, this one can't be. It's not like you can put an N and then have more P's over here. So yes, one could look, does A go with A? Well, no, but also it doesn't matter because you wouldn't get another P nucleotide when you already got an N. And so for those last two, I would write N. And that would be it for that kind of question. Do you want me to do another one of those, or do you feel like you get it and you want to move on to other stuff? Yep.
I think I'm just confused. Like, that makes sense, but the process of how the P and the N got made, I'm confused on.
Okay.
Once the base pairs are put back in, and the P nucleotide, they're just complementary to the original.
Yep. Yeah. Okay. It's just because it was originally like this, like you had one strand and a second strand, and then because the strand got cut here, the strand went like this. Whoop. So the things that originally were complements now came next to the other strand.
I think I got confused when we said the palindrome. So we said same forward and same backwards.
Yeah, so here it really would look same forward and backwards if I drew both strands. Or honestly, the P is the same as AT, and there'd be AT. That's not a very long palindrome.
So palindrome is whatever the other stuff.
Yeah. Okay. So here's the other one, if you want it. So, and you can see, once upon a time, somebody wrote me this whole long thing about how do I know they're not all N's? And they went through this whole thing about they were all being N's, and I was like, technically you're right. So now I wrote, if it could be a P, you have to write P. So you can see I learn over time. So again, what's the first thing I'm going to do here? Yep. Okay. All right, so this. Yep. Okay, now what? Okay, now we got 4 base pairs. So what do we think? Okay, so G cannot be a P nucleotide on the left, right? Once you are on a side and you find one that's not a P nucleotide, you're done with that side. All right, so let's go to the other side.
Oh, it wouldn't be the G next to it. It wouldn't be.
No, because this is part of that whole, it happens P nucleotides and then N. So you can't have an N before a P. So basically you look at each side, say, is it a P or an N? If once you've stopped getting P's, you're done for that side. I guess technically, if you got more than 5, you also would be done, but I don't think I've ever drawn one that's that long. Yeah. This G?
Yeah.
Because you would look and say, can G bind to A? Does G pair with A? Could this have earlier been the base pair on this other strand that was bound to the A?
Oh, so you're like thinking back to that.
Exactly. Could it have been a P nucleotide? Since we know how P nucleotides form, is it possible that formed through P nucleotide addition? No, because then if the way that it would have formed is if it would have been base paired to this A, and G doesn't go with A. If it was a T, then you would say yes. Okay, but it's not. It's a G, so you look and you say, nope, that's not a P nucleotide. Yep, Brooke. So we're gonna look over here and we're gonna say this one's not a P. So let's look over here and check for the other side. Okay. So now we start by checking on this T. What's the deal with the T? It could go with the A. So what does that make it? So that makes it a P. So we're gonna say that one's a P. Then what's the next? Then what do we think about the next base pair? It's an A. Who would the A have gone with? This T. Could A go with T? Yeah, so that could be a P nucleotide. All right. What about the G? So the G we're testing, could it go with the T? No. So we're done with P nucleotides on that side. We did both of our P nucleotides, so then everybody who's left over, and so that would be how it would work for this one. So this is one of the types of questions that the first time people see it, they're like, ah, and then afterwards they're like, please ask one like that on the exam because I know how to do it. So we need to talk again about some more details of VDJ recombination. A couple of pieces that I want to briefly just tell you about that are related to thinking about VDJ recombination. We've talked a lot, and it's sort of inherent in the name what's going on with the V, D, and J segments. I really haven't said a ton about what's going on with the constant region DNA. And so I have a couple of things I want to tell you about the constant region now in terms of what's going on between, say, what happens with the DNA and how we get to the mRNA and eventually the protein. So what goes on with this constant region stuff. As you can see, the constant region doesn't get touched when we do the rearrangement of the DNA in terms of particularly kind of like this upstream constant region. But if we look at the locus, so here you can see the V's, D's, and J's, and then you can see the different constant region DNA here. And you can imagine once we have done rearrangement and we've put together a V, D, and a J, now we can actually start doing transcription there and transcribe that region. So we're going to transcribe from there on. As you could see in the earlier slide, you transcribe everybody, then you splice out the RNA sort of that you didn't need. So those extra downstream J's, for example, get spliced out. But then you can also think about that same RNA polymerase who transcribed the V, D, and J, and it's going to keep on going to transcribe constant region exons. And I want you to look at this image at the top where you can see the chromosome and some of the exons for the constant region. So you can see that they are in that MDs give everyone apples order. But what do you notice about the distance of the DNA between all of them? Yeah, Jonah. Where are they far apart? Are they equally all far apart? No. What's unique? Like where do things sort of change up? Around gamma. So M and D, or mu and delta, are relatively close to the VG and J segment, and then there's a long way to gamma. And there's a pretty long way to each of the rest of them. So, 6.5, 4.5, and then suddenly 55 more before you get to the next one. So if you're the RNA polymerase, you're going along doing RNA polymerase, do you think you're going to make it to M or to mu? Yeah, that's not too far. You're not tired yet. You make it to D? Yeah, that'll work. You going to make it to the first G? No, that's way too far. You're way too tired. And so the RNA polymerase really only transcribes M and D and doesn't transcribe the other ones. And it's because they're just too freaking far away. Generally, you can see the RNA polymerase will transcribe the VDJ region and will transcribe M and D, the M and D regions. And so the RNA, at least the original RNA, will have both the M and D information. And then based on RNA splicing, the cell will either make M or make D. And because this is something that's happening at the level of RNA, the cell can sort of make some M, make some D, be kind of switching back and forth. It's not totally committed, can do either. But as you can see, we're not getting those other kinds of isotypes. Similarly, we talked about the fact that the B cell receptor is really just an antibody with a transmembrane domain. When we actually look at the exons, so here you can see the actual DNA that makes up mu. You can see there's actually a few different parts. There's a part that's basically like the end, or there's a transmembrane domain. And so the RNA can actually have both, and we can use alternative splicing to make a version without a transmembrane domain and a version with a transmembrane domain. And so again, the B cell is going to make this change by changing up the RNA. It's going to originally have an RNA that has all the choices, and then change by RNA splicing. So it's not committed to either one. It can actually do both at the same time. All works pretty easily. So we talked about how amazing VDJ recombination is, what happens in an organism who cannot do VDJ recombination. But we also said that if Dr. Dunaway was here, he would mention something about you getting dead and DNA breaks. And I told you about all the ways that VDJ recombination can go wrong. And it goes wrong sometimes. And so what we have to think a little bit about from the process we saw on Friday is, so how do we limit that? What sort of safeguards are in place so that these types of recombination errors don't happen? And there are two particularly big things that happen to limit the recombination errors. One of them, your textbook actually told you, but didn't like tell you it was telling you, in this figure, where you can see your textbook basically says RAG and just draws it as one big oval, one big blue oval. And the reason why that actually makes sense is that all of the enzymes that we've talked about, RAG1, RAG2, Artemis, all bind to each other and act as one big complex. So they all kind of travel in a pack. Why do you think it might be important for all of those enzymes to be bound together and act as a pack? So the idea is they're all acting on the same area. The only DNA that we're messing with, the only DNA that's getting broken is the DNA in this region with the RSSs. The rest of the DNA is not, it's not like one enzyme is cutting over here and one enzyme's cutting over here, and then you're like, look, I have all these double-strand breaks, let me fuse them together. Yeah, that would be bad. Instead, the enzymes are all working at the same place, and so they're only cutting in the same location. And you can see they also kind of have pockets that hold the DNA ends. So once that DNA is broken, it doesn't like float away to join to some other chromosome. That DNA is actually held in that enzyme and isn't allowed to go invade other places. If you look back at how I drew this sequence, you see how I lined up the two RSSs together? That's actually how the DNA is held. And you have the enzyme sort of holding the two RSSs together and having all of these reactions happening right there. So the DNA is not going to like go invade some other chromosome because it's getting held right in that one location. There's one other really big reason, or a really big part of how we limit recombination errors. And this question, you will see, I think I asked a question about on the problem set or something similar to this. I've asked questions about it on old exams. And every so often, someone, instead of giving me a mechanistic answer, gives me like a philosophical answer to this question. And so I will tell you I meant a mechanistic answer. But the basic idea here is that if you think about your genome, you think about the DNA that encodes proteins. This blew my mind in high school bio, so we'll use this example. Every gene in my entire genome has or every cell in my whole body has the genetic information to make rods and cones and eyes. But my whole body isn't eyes. Only these cells chose to make rods and cones, right? The other ones are just like not using that DNA. We regulate expression of when we actually transcribe certain genes and translate certain proteins. Same thing with RAG1 and RAG2. Every cell of your body has the genes for RAG1 and RAG2, but they don't transcribe those RNAs and they don't make the proteins. Those two, the making and formation, and I don't know what that word was going to become, the making of the RAG proteins is under super, super heavy regulation. You can think of these enzymes that are nicking and cutting DNA as being a little dangerous. And so we don't let just any old cell transcribe and translate these enzymes. In a B cell, for example, that B cell is allowed to turn on RAG twice in its life. And that's it. So you can see there's one stage where we turn on RAG, and then we turn it off for a while. And then there's one other stage where we turn on RAG, and then it's off. And then when it's off for the rest of that B cell's life. Because we don't want mistakes where we are damaging the DNA. The only cells in your entire body that make the RAG proteins are B cells during these two phases of their life and T cells during the two phases of their life. Because we tightly regulate the expression so that RAG can't damage DNA in any other part of this. Some of the other enzymes that are involved, like TDT, like Artemis, are not as tightly regulated. Some of them have other roles in DNA repair. So Artemis actually can do some other DNA repair stuff. So they can be on all the time, but the cutter cannot be on all the time. The cutter is on 2 times and only 2 times, and that is it. So right now, we're still in this phase of the B cell's life, where the B cell is undergoing sort of this generation of diversity. Each B cell is making a unique receptor, and we're in the primary lymphoid organ. You should think of this as the time before the B cell sees antigen. So we're still in an antigen-independent phase. We are here. There are other phases that happen later in the B cell's life after we see antigen. And so this was the slide you guys got super excited about before, and I was excited that you were excited about, you already have one B cell against every epitope ever, because you'd made them in your bone marrow in this like specialized compartment away from the antigens, and then they left and are trying to find their antigens elsewhere. Later on in the B cell's life, some other changes can happen to the genes that encode the antibody. Those changes include changes to the variable region so that we can get tighter binding to antigen. And those changes also happen with the class switch, where we can get the change in which isotype. And that class switch is what lets us use things like IgG, IgE, or IgA and deal with that 55 kb gap that I showed you. Those are both things that happen late in the response. Those are things we are not dealing with right now. They happen after you see antigen. One thing that you might also be able to realize because of what I've been telling you is that these happen using enzymes other than RAG. Because RAG, when we stop B cell development, RAG's off, and RAG's off forever. So these don't require RAG, they require other processes. So these are not things that we're going to really be dealing with right now. But what I do want you to note is if you look at the name of one of them, it's somatic hypermutation. And that's the name of the process that allows us to change the affinity. What's the very end of that phrase, somatic hypermutation? The last, Mutation. When you hear mutation, what do you think about? You think about DNA and permanent change, right? That process of somatic hypermutation is going to be another DNA situation. And so it's going to be a permanent change to the B cell, but it's a permanent change that happens later in the B cell's life. Similarly, the class switch is actually called class switch recombination. It is another change that's happening to the DNA. So it's another permanent change that happens to that B cell later in the B cell's life. And so here you can kind of see a big picture summary of all of the ways that those immunoglobulin genes can change. We can recombine the DNA through VDJ recombination. Note that that is irreversible. We permanently change the DNA. You can't go back. We added some PNN nucleotides. That's a change to the DNA. You can't go back. We brought the promoter and enhancer closer together because we were throwing out the extra DNA. That's a change to the DNA. Can't go back. We can actually make IgM and IgD by RNA splicing. So that when we can go back, we can go between M and D and M and D and M and D. We can go from transmembrane antibody to secreted antibody by whether or not we have that last part of the gene in the RNA. That's an RNA thing, so we can go back and forth. We can make both. And then later on, we're going to see that change in affinity of somatic hypermutation. We're going to see class switch. And again, those are going to be DNA processes and are going to be irreversible. You can't go back. So this table, I'm actually covering up part of the table because this table was really meant by this other textbook to compare and contrast details in B cells and T cells. But here you can see the B cell answers. So you can see, do we use VD and Js? Yes. Do we have a light chain? Yes, it's either kappa or lambda. Do we have a heavy chain? Yeah, V's, D's, and J's. Do we use RAG? Yup. Do we have P and N nucleotides? Yup. Can we do things like put D with D? No, because the 1223 rule doesn't let that happen. Do we do allelic exclusion, which will be coming up later? Yes. Do we actually make a secreted product, like the secreted antibody? Yes. And then, can we have things like somatic hypermutation? Can we think about some of the details of the consonant region? Yes. Again, those are going to be things that are different in T cells. So this is just sort of an overview of what you've seen. So what we need to do now is sort of take this process of VDJ recombination and have it not just be kind of happening in the void, but now actually have it happening in a B cell. And I'll think about how the B cell sort of regulates and deals with this process while it is developing in the bone marrow. I will also, just because I have to feel like a complete immunologist, will tell you that I am actually officially, when I'm saying B cells here, I am actually talking about something called a B2 B cell, which is sort of the classic conventional kind of B cells. There are other weird cells. So there is, for example, this other weird cell called the B1 B cell. I will say a little bit more about B1 B cells and sort of the other weird lymphocytes, which is the easiest way to put them, later on. They actually are these weird lymphocytes that kind of have some features of the innate immune system and kind of have some adaptive features. We're not going to deal with them right now. We're just officially talking about B2 B cells, which are like the classic version. But yes, there are other types that I am ignoring. And so to think about B cell development, we have to kind of start with thinking about hematopoiesis in general. So if you remember, we started out hematopoiesis with the hematopoietic stem cell, or the HSC, from the bone marrow. And that cell could get signals that could either make it go down the lymphoid path or down the myeloid path to decide to become either a lymphocyte or a myeloid cell. And further signals can push the cell down many of these different paths. And you can see after that cell decides to be a lymphoid cell, it will make further decisions, some of which could involve becoming a B cell, which is really what we're thinking about here. By and large, hematopoiesis is happening in the bone marrow. You can see that it starts happening in the bone marrow in the fetus at around 4 months and really kind of gets to peak at birth, but then stays relatively high. So you are doing hematopoiesis in your bone marrow throughout life. In some earlier stages, hematopoiesis is happening in some other locations. In the embryo and fetus, we do see some hematopoiesis happening in the yolk sac and the fetal liver and spleen. And so the point that I want you to see is that the fetus has a lot of these cells that are kind of almost ready to go at birth. And then for the rest of your life, all of us, wherever we are on this line, I'm not going to feel sad about being so very different from you on this line. But we're all still kind of doing pretty similar amounts of hematopoiesis with our bone marrow. This is actually the point of bone marrow transplantation. If you are getting a bone marrow transplant, what we are doing is we are taking bone marrow from one person and giving it to this other person, because you, the recipient, had a hematopoiesis problem. There's something that's an issue with hematopoiesis for you. We can think about lots of reasons why that might be. One reason might be that we gave you chemotherapy or radiation that killed all your stem cells. So now we're going to give you some of somebody else's stem cells. And this will allow us to basically regrow an immune system and to restore hematopoiesis, have it rehappening. And so you can see here's a situation where one has a mouse. that mouse gets radiation. Now, basically, the mouse, all of the rapidly dividing cells in that mouse are killed, which means all of the immune cells are killed. This mouse basically has no immune system. In a slide that I will show you later in the semester where they are trying to tell you about radiation, they show a person before and they are like shaded, and then after they show them blank because like we've wiped out their immune system. And what we can do with our mouse is give our mouse some bone marrow cells. You can see that the bone marrow actually has a mixture of a lot of cells. So we've got some eosinophils and some erythrocytes and some lymphocytes and a bunch of stuff and some stem cells. There's the one little stem cell right there. And we can give that bone marrow to the mouse, and the mouse will live happily ever after. One thing, I have spent a lot of time doing some bone marrow transplants at one point in my life. And one thing that always bugs me about this slide and the next slide is that they show you're putting it into like the mouse's back. That is so not correct. You actually have to basically give the mouse an IV. You actually have to inject into a vein. You can imagine that that's tricky, right? Sort of difficult to do an intravenous injection on a mouse. That actually I'm gonna mention that on the next slide, but know that this is actually a pretty like a thing you got to practice before you can actually pull off this injection well. And so what this paper shows is the strength of that hematopoietic stem cell. And so what they did in this paper is they said, I don't feel like using a whole mixture of all of these kinds of cells from bone marrow. Instead, I'm going to try to purify just hematopoietic stem cells. And they wanted to know, how many stem cells do you need to grow an immune system? If I got rid of your whole immune system right now, and I was giving you back hematopoietic stem cells, how many would it take to grow a whole immune system? Is really what they're thinking about here. It's kind of thinking about how powerful is that hematopoietic stem cell. And so at one point, they gave mice 20 hematopoietic stem cells. And 100% of the time, that gave them a whole immune system. That's pretty cool. They tried to give them 10. 90% of the time, that worked. They tried 5, 2, and even 1. And you can see that when they just gave them 1 hematopoietic stem cell, 20% of the time, that was enough to grow back an entire immune system. So you only need 1. And when I look at these numbers, I think about how hard that injection is. And I think about the fact that probably, in my mind, I bet 1 actually works. It's just that you messed up the injection 80% of the time. And so this is how frequently you actually pulled off the injection perfectly of that 1 cell. And really, you can see that a very small number of hematopoietic stem cells are enough to reconstitute your entire immune system. So thank you, hematopoietic stem cells. Since we're talking about B-cell development, we have to remember that we are in a primary lymphoid organ. And specifically, as sort of I have implied here, we are in the bone marrow for our primary lymphoid organ. So the HSC is in the bone marrow, and it stays in the bone marrow for this entire process of B-cell development. So we are totally in bone marrow. Here you can see that happening. And so the HSC, all of the stuff that you see up here, like HSC and on, that's all bone marrow stuff. And pretty much all the steps to get to the B cell also happening in the bone marrow. And so you can see this happening in the bone marrow. Notice that this shows the bone marrow as being antigen independent. If I wanted to argue with that, I could. But let's pretend that we don't argue with that. Let's say that this is like the clean room where there's no antigens and you're not totally worrying about antigens here. You're just doing VDJ recombination. And then after B-cell development, after the B-cell has finished all the steps that it's going to do today and on Wednesday, it gets to leave the bone marrow and go to the rest of the body where it now is going to be seeing antigens. And what you should notice is that we are all making about 5 million new B-cells a day. So there's about 5 million new B cells a day coming out of our bone marrow every day. one of the things that we have to think a little bit about is why bone marrow? What is important about the bone marrow for this process? And there are two big things that are really important for us thinking about the bone marrow. One of them, again, I will say, is you can kind of imagine not so much antigen. So again, we're going to imagine it at least as the clean room, at least for now. So, imagine the lack of antigen. But then there's a second thing that's really important about the bone marrow. And to sort of make this second thing really clear, I want to tell you about developing lymphocytes in general. This is true of B cells throughout development. This is true of T cells throughout the development. Those cells developing lymphocytes are exceptionally needy. I want you to think about the neediest person you have ever met who constantly needs you to tell them they're doing good. You're doing a good job, it's okay. You're doing a good job, it's okay. Developing lymphocytes are needier than that. Basically, developing lymphocytes, if they're not getting a signal, a little pat on the head, good job developing lymphocyte, good job developing lymphocyte, they die. They really want to die unless they are constantly getting their little pat on the head signal. And so the really nice thing about bone marrow is there are lots and lots of signals in bone marrow to keep developing lymphocytes alive. And so you can see those cells of the bone marrow, the structural cells, are making all sorts of factors that can keep those developing B cells and the developing T cells when we get there alive. One of the most famous ones is one called IL-7, which is a cytokine. It's a survival cytokine. So basically IL-7 makes cells survive, particularly lymphocytes. And so here you can see all these different shades of green are B cells developing in the bone marrow. And you can see that the location of the bone marrow is actually quite important because that developing B cell is making all sorts of contact with bone marrow cells who are giving it its little pats on the head and keeping it alive. Otherwise, it would try to die. And the only one I'm going to call out here is IL-7. And so here you can see, look, IL-7, it signals through an IL-7 receptor. And it does things like make the cell survive, which is the main function of IL-7, though it also can impact a lot of other parts of B cell biology. And so it's really important that this happens in the bone marrow. If a developing B cell escaped randomly, if I ever say a cell escapes, I mean it got out of a place where it shouldn't have. I asked about a cell escaping on an exam once, and it threw someone off. They're like, what do you mean? And I'm like, no, escaped. It means it wasn't supposed to leave, but it did. So the idea, if my developing B cell escaped from the bone marrow, it would be out of luck because it wouldn't get any IL-7, and it would be sad, and it would die. So these steps that we're thinking about are all of these bone marrow steps that are in yellow here. Other steps like actually binding to antigen or doing things like somatic hypermutation, class switch, those are all later after bone marrow. Those are all the pink things. Not our problem now. We're only in the yellow stuff now. And the other piece of this that we need to kind of think a little bit about is activating a B cell. The reason why we have to think about this is that I just told you previously that B cells and lymphocytes, particularly developing B cells, are really needy. They need a lot of signals. And one of the things that we're going to have to see are some ways that we give the B cell signals as those B cells develop. And so you need to know a little bit about B cell signals to think about B cell development. The most important signal once the B cell starts developing is a signal that will come through the B cell receptor. Usually pretty easy to remember that the receptor on B cells is the B cell receptor. And you can see the B cell receptor on the left. You can see it's really just an antibody with a transmembrane domain. And so you could imagine that making a B cell receptor so that you can get a signal is really the same as making antibodies, like we've just said. And so as I talk about the rest of B cell development, I'm really going to be talking about making the BCR, but it's the same thing as making the antibody, because it's actually the same, even RNA that's just spliced differently. So this is my very broad view of signal transduction. I could talk about signal transduction all day. I really like signal transduction. But here I have a specific kind of point I want to make about signal transduction. This image shows general signal transduction with two different kinds of receptors. I'm mostly going to talk about the one that's on the top. Yes, there are variations like the one that's on the bottom. The one that's on the top is pretty much what we often think about. And often we talk about signal transduction with something called receptor tyrosine kinases, or RTKs. And so you can think of the top as an RTK. So insulin receptor, a lot of our cytokine, like so many RTKs. The key thing I want you to know about an RTK, which is true of most all receptors in signal transduction, is that an RTK, or receptor tyrosine kinase, has three parts. If you had to guess from the name, what would the three parts of the receptor tyrosine kinase be? Yes, David. So the three parts of a receptor tyrosine kinase are the receptor, the tyrosine, and the kinase. The receptor is the part that's outside the cell that is binding to ligand. And so here you can see this sort of, I don't know what, quadrilaterally part that binds to the red triangle ligand. What you can notice, and this is pretty common among a lot of RTKs, is that you can actually see that this receptor is in two parts. Those two parts are actually diffusing through the membrane separately from another. So they're wherever in the membrane, but they're not usually together. They're just like around. And if the ligand happens to come by, they both grab the ligand, and now they're next to each other. Whereas before they were not next to each other. So you can see how they get pulled close together because they've bound ligand. It's actually known as induced proximity. They have been induced into proximity of each other. The ligand is binding to either part of the receptor, and that's making these two receptor chains be next to each other. And specifically, when you think about that, and when you look at that picture, you see that you have taken these two receptor chains and pulled their extracellular domains next to each other. When you pull the extracellular domains next to each other, what happens by default? Yes, David. The intracellular portions come together. It's not like the tops come together and the bottoms stay apart. The whole thing gets pulled together, right? So you can see here, not only did the extracellular portions come together, the intracellular portions came together. The intracellular portions contain two parts. One of them is the tyrosine and one of them is the kinase. What is a kinase? You guys have heard about kinases from Dr. Donnelly. What's a kinase? It is in fact an enzyme. It has ACE at the end, so we know it's an enzyme. Yes. What does this enzyme do? Yeah, Dave. It adds phosphates to things. So kinases add phosphates to things. It likes to add phosphates to things. One of the things that kinases most like to add phosphates to are tyrosine amino acids. And so what happens is that you've got this kinase domain. It's normally like floating around the cell, doing whatever. It's not next to anything, so it can't actually like add phosphates to things because it would need to be next to it to add phosphates. And when it gets pulled next to this other protein, because of the induced proximity that came from the ligand, now the kinase domains get shoved next to each other where they weren't before. And now they can phosphorylate each other. So they both have a place to be phosphorylated, a tyrosine, and they both have an ability to do phosphorylating, a kinase. And so they phosphorylate each other, which is shown here as these two dots being added, these two pink dots being added. And now stuff happens because of those pink dots having been added. That's sort of what we need to think about for signal transduction right now. The only reason I leave the bottom part of this here is because what you can also see is that there are situations where all the parts are not done by one protein. Sometimes you actually have multiple proteins interacting, like here the blue one and the green one, to make all this work. But it's really the same process pretty much has to happen for just about all kinds of signal transduction. So there's the B cell receptor. And in this image, it's a pretty good picture of what I want to show you, the B cell receptor. I'm showing you this image very much on purpose. I want you to look at the B cell receptor and compare it to what I just told you about receptor tyrosine kinases, which I will tell you are like the classic signal transduction. How do you feel about the B cell receptor compared to a receptor tyrosine kinase? What sort of compare and contrast might you think of? Yeah, Jonah.
I guess in this case, there's only one binding site to the antigen compared to the two sites coming together.
Okay, so you're like, well, so you're a little worried about binding sites. We can come back to that because there's a bigger worry.
Only one binding site binds to one antigen.
Okay. There's another big issue here. Yeah, Jonah. Yeah, look at where the B cell receptor ends. Remember how I showed you all these things that were happening with the like intracellular tail of the B cell receptor? Or of a receptor tyrosine kinase? I picked this picture because it actually shows you correctly how little of the B cell receptor is inside the cell. So yeah, we have like awesome receptor part that we did VDJ recombination to make, but there's no tyrosine and there's no kinase because there's really no tail inside the cell. So that seems kind of crappy. So the B cell receptor as a receptor kind of sucks. And so the B cell receptor only works when it has help from a lot of other proteins. And two of these key proteins that the B cell receptor needs are called Ig alpha and Ig beta. They are also known as CD79A and CD79B. So you can either call them Ig alpha or Ig beta or CD79A and CD79B. And you can see that these proteins actually don't really have much in the way of an extracellular domain. They're not going to bind any ligand, but they have a really nice intracellular tail. And through some really cool biochemistry, these actually travel together. So even though they're just separate proteins, they actually diffuse through the membrane together. They always are next to each other. And so the B cell receptor has to have these co-receptors that are going to help it signal. They specifically provide this domain called an ITAM. The only real thing you need to worry about with the ITAM is, you know what T in ITAM stands for? No. What did T stand for in RTK? Tyrosine. Also tyrosine. Basically, now this extra friend that the B cell receptor has, Ig alpha and Ig beta, that provides the tyrosine. So now this can actually like do a signal. Because before, without this friend, it can't actually do a signal. Yes, Brenna.
Just to clarify,
Yeah.
So the mechanism that we were talking before was for the signal transduction.
It's for signal transduction of a lot of other kinds of receptors, lots of other stuff, but it shows us kind of what you need to do signal transduction. Yep. What I'm not showing you here is the kinase. You're going to have to just take my word for it that there's a kinase. The reason why you have to take my word for it that there's a kinase, I thought I had it. The reason why you have to take my word for it because if there's a kinase is that there are actually different kinases in different situations. And so I'm not going to go through all of the different options of the kinase now, but just say there's a kinase. And so the way that this will work, and this sort of answers the point that you were making, Jonah, is that usually what has to happen is you usually will have multiple B cell receptors that bind antigen that come together and have induced proximity and have some of those kinases acting on the tyrosine in that Ig alpha and Ig beta. And so typically we're going to have multiple B cell receptors on one B cell, get cross-linked, pull all this stuff into proximity, and get some signaling is usually how it's going to work.
And we said, I'm sorry.
Yeah.
I thought the IgA and IgB were floating or with it.
They stay floating next to it. They don't float separately. They stay floating next to it. That's super fun biochemistry. Actually, somebody I went to grad school with proved that. It's really cool. And now it's like in all the textbooks, and like his grad school work is like in all the textbooks. So yes, they float together because of cool biochemistry that Matt did. And then eventually you get signals happening in the cell. There are a couple of things I want to tell you, tell you here, reasons why I'm showing you all this signaling. One thing that you can see will happen is that we're going to change gene expression at the end. So we're going to start doing transcription of new stuff that we weren't doing before. We also might see some functional changes in the cell, like some cytoskeleton changes, or most importantly for us, the cell might start to divide. So we're going to have the cell potentially go to mitosis in some cases. We're also going to see the cell get some different functions, like that will happen with differentiation and activation. And so all of these are kinds of things that will happen to the B cell once it gets a signal through its B cell receptor. The other reason I'm showing you this, you do not need to care about any of these proteins that are happening down in this downstream signaling cascade. When we get to T cells, we are going to learn about the proteins in the downstream signaling cascade. When we learn about T cells, come back to this slide and look at it. You're going to be like, wait a minute, it's all the same themes. And in fact, there are a lot of ways that it's really similar. It's just that we're not dealing with that right now. That's a later problem. And so it actually does look quite similar to signaling downstream of the T cell receptor. And here you can see there are other proteins that can interact, lots of different versions. Many of them may bring the kinase and help this process out. One of them you actually already know about, it's a complement receptor. So if complement has bound to stuff, then the B cell will work better. If it binds to an antigen that has complement on it, then it gets more signals, more pats on the head, and works even better than it might have before. So we're going to start with a number of the steps of exactly how we get to B cells. on Wednesday. So I will see you guys then. Remember that your problem set is due on Wednesday as well. And be sure to come by if you have questions.